Hey everyone, welcome back. Today we're going to do a deep dive into peptidoglycan.
PG.
Short and sweet, right? You know, it's essential, but
It is.
but how well do you really understand this structure?
Yeah.
We're going to be taking some insights from a book by Walsh and Vencevich called Antibiotics: Challenges, Mechanisms, Opportunities.
Great book.
And by the end, you won't just have a really solid understanding of peptidoglycan, but we'll also give you 5 exam questions to test yourself on.
Yeah, it's really critical for medical microbiologists, getting this PG stuff down pat.
It unlocks so much, right, about how bacteria work and how they interact with us.
Definitely.
So let's just start with the basics. Why is PG so important for our bacterial buddies?
Well, just imagine a bacteria without its PG layer.
Oh, no.
It would be like a water balloon. Without the balloon, poof.
Yeah.
So the PG, it's like an exoskeleton.
Okay.
A rigid but dynamic structure.
Okay.
And it protects the cell from all that pressure inside.
That osmotic pressure.
Osmotic pressure.
Yeah. Nobody wants to be a puddle.
Nope.
So PG is like a pressure suit.
It is a pressure suit, but it's also more than just protection from bursting.
Right.
Because bacteria face all these external threats too, right?
Yeah, of course.
Immune cells, harsh environments, even other bacteria.
They're fighting for those resources.
Exactly.
Yeah, it's a tough world out there.
It is.
So PG is that first line of defense against all that.
It is.
I like to picture it like a knight in shining armor, ready for battle.
Yeah.
But, bacteria aren't static.
Right.
They're constantly growing and dividing.
Right.
How does the PG keep up with that?
That's what's so cool about it. It's not static.
Okay.
It's constantly being remodeled, broken down, and rebuilt as the bacterium grows.
Okay.
And for you guys as medical microbiologists,
Yes.
understanding this is key to understanding how antibiotics work.
Okay, so let's break down that structure because I always get that a little mixed up.
Okay, so think of it like a chain link fence.
Okay, chain link fence, yeah.
Surrounding the bacteria.
I can picture that.
All right, so the vertical supports, those are the glycan strands.
Okay.
Long chains of sugar molecules.
Sugars.
We're talking N-acetylglucosamine, GLC-NAC,
GLC-NAC.
and N-acetylnoramic acid, or MRNAC.
MRNAC.
And they're linked by a beta-1,4 bond.
GLC-NAC and MRNAC, the dynamic duo.
Yeah.
Okay, but what about those horizontal wires
Yes.
of the fence?
Yes, those are really key for strength.
Yeah, those are the peptide crosslinks, right?
You got it.
Okay, so those are the rivets.
The rivets holding it all together.
Okay.
Here's a really key difference for you to remember.
Okay.
The composition of these peptide crosslinks.
Okay.
It varies between gram-negative and gram-positive bacteria.
The gram stain,
Exactly.
the foundation of bacterial ID.
So in gram-negative bacteria, you usually find diminopamelic acid,
DAP.
DIP, at a key position in the peptide chain.
So DAP is like the gram-negative signature.
You could say that, yeah. But in gram-positive bacteria, it's usually lysine in that position.
Okay.
And often they have these bridges of glycine molecules,
Interesting.
making the crosslinks longer and more flexible.
So that's why gram-positive bacteria have that thicker PG layer.
Exactly.
Because they've got those extra glycine bridges.
And this difference in cross-linking is why certain antibiotics, like vancomycin, are more effective against gram-positives.
Right.
They can target those specific crosslinks.
That makes so much more sense now.
Good.
I can picture it.
Good.
But wait a minute, there's more to the story, right?
There is.
It's not just those two components.
So what's fascinating is other molecules can attach to this scaffold,
Okay.
like proteins and teichoic acids.
Really?
And these attachments, they're not just decorations.
Yeah.
They give the bacteria unique properties.
Oh, wow.
They can even change how they interact with their host.
What kind of attachments?
Well, in gram-negative bacteria, there's bronze lipoprotein.
Bronze lipoprotein.
It's covalently linked to those peptide chains.
Okay.
And it helps anchor the outer membrane to the PG layer.
Okay.
You know how some gram-negative infections are notoriously tricky to treat?
Yeah.
Well, bronze lipoprotein is part of that.
What?
It makes it harder for some antibiotics to reach their targets.
So it's like a bodyguard for the cell.
It is. It's a bodyguard.
Wow.
And then in gram-positive bacteria, there's protein A.
Didn't we learn about protein A in that Staph aureus lecture?
Yes.
Okay.
It's a key virulence factor for Staph aureus.
Right.
It binds to antibodies and it flips them.
What?
Yeah, so it hides from the immune system.
Sneaky.
Sneaky bacteria.
So these attachments aren't just structural.
Right.
They can play a role in virulence.
Absolutely.
This is why we have to understand the connections.
Exactly.
Not just memorize facts.
Right.
Okay, so here's another layer.
Okay.
The PG layer itself influences the shape of a bacteria.
Yeah.
So like whether it's a rod or coccus or something in between, it acts like a mold, giving the bacteria its form.
Yeah, think of it like a balloon animal artist.
Okay, yeah, I like that.
Shaping the PG layer.
To create all those different shapes.
Yes, exactly.
Wow.
So to create these shapes, bacteria have to really carefully orchestrate the synthesis and degradation of PG.
Okay.
They use these cytoskeletal elements, like internal scaffolding, to control where and how the PG grows.
It's a dynamic process.
It is.
That's how they maintain their shape as they grow and divide.
Yeah, so it's not just building the wall. Yeah. It's shaping it precisely.
Like bacterial origami.
Yes.
Wow.
And for you guys as medical microbiologists, understanding this opens up possibilities for developing new antibacterial strategies. Yeah. Wow. Because if we can mess with how they build their PG, we can weaken their defenses.
So we've talked about the big picture of PG structure and why it's important, but how is it actually built?
That's where things get really fascinating.
Okay.
It's a 3-phase journey.
Oh, wow.
Starting deep inside the cell in the cytoplasm.
Okay.
Think of it like a factory.
Oh, a factory.
Yeah.
Okay, I'm ready for a tour. But before we get lost in all the details, let's take a break. And then when we come back, we'll dive into that first phase of PG biosynthesis.
Okay, sounds good. So we're back in the bacterial factory, ready to see how those PG building blocks are made.
I'm ready for the tour. I love a good factory tour.
So picture the cytoplasm as this bustling workshop where all the materials are gathered to build the PG.
I can picture the little molecular carpenters.
Yeah, getting everything ready.
So what are the materials?
Well, it all starts with glucose.
Oh, glucose.
You know, the basic sugar.
The building block of life, right?
That's right, but the bacteria use it to build PG.
From something so simple to something so complex, amazing.
I know, it's microbiology.
Right.
So they take that glucose, and through a bunch of steps, they convert it to GLCNAC1 phosphate.
GLCNAC.
Remember GLCNAC, one half of our dynamic duo.
Right, from our glycan strands.
Exactly.
Okay.
So then this enzyme, MIRA,
great.
it attaches a 3-carbon unit to that GLCNAC.
A 3-carbon unit, where does that come from?
It uses phosphenylpyruvate, or PP.
PEP, wait, isn't that involving glycolysis?
It is.
So they're multitasking.
They're efficient little buggers.
I know.
So MIRA does its thing, and we end up with UDP-MIRNAC.
UDP-MIRNAC.
The foundation of our PG building block.
Okay, UDP is uridine diphosphate, right?
Yes, it's like a carrier molecule in a lot of these biosynthetic pathways.
So UDP-MIRNAC is like the base of our house.
Right.
What about the walls and the roof?
The pentapeptide.
Yeah.
Remember those peptide crosslinks?
The rivets of our fence.
Exactly. So those are built from pentapeptides.
Pentapeptides mean?
Chains of 5 amino acids.
Okay.
And they're assembled 1 amino acid at a time.
So like a little assembly line?
It is an assembly line.
Okay.
With these enzymes, MERC, MIR, and MERF.
What a team.
Each one adds its amino acid to the growing chain.
Oh, wow. So organized.
And this process takes energy, ATP.
Okay.
Bacteria, they really invest to make this PG.
It's worth it.
It is.
And they know it.
Here's a key detail.
Okay.
The last 2 amino acids in that chain, they're dealanines.
Dealanines. Why is that important?
Well, most amino acids in proteins, they're in the L form.
Okay.
But in PG, we see D amino acids.
Interesting.
And it's crucial for how it works.
And probably for antibiotics, too.
You're thinking like a medical microbiologist.
I try.
These dealanines are supplied by 2 enzymes.
Okay, what are they?
Alanine racemose.
Alanine racemose?
And dealloligase.
Okay.
And these are targets for some antibiotics, like glycerine.
Okay.
It inhibits both of those enzymes.
Oh, wow.
So no dealanines.
No PG.
Exactly.
Okay, so we've got our UDP-MIRNAC pentapeptide building block.
We do.
All ready to go, but it's still stuck inside the cell.
It is.
We got to get it to the membrane.
We do. That's phase 2.
Phase 2, the membrane phase.
Okay. So how do we get these building blocks across?
Yeah, that membrane is a tough barrier.
It is. So we have a special lipid carrier.
What is that?
Bactoprenol.
Bactoprenol.
Okay. It shuttles the PG building block across.
From the cytoplasm to the outside.
Exactly.
Bactoprenol sounds like something out of Star Wars.
It kind of is. It's remarkable. Yeah. It's this long chain lipid with a head that can attach to different molecules.
I'm picturing like a tiny submarine.
Oh, I like that.
Ready to transport our cargo.
Perfect.
Oh, yeah.
And the tail is hydrophobic.
Hydrophobic meaning?
Hates water. loves fats.
Okay, and the membrane's made of fat.
So it embeds itself right in there.
So bactoprenol has one foot in the membrane and one in the cytoplasm.
A very strategic position.
Ready to grab our building block.
Exactly.
Let's get away.
So first, we form bactoprenol diphosphate. We link 8 isopentenyl pyrophosphate molecules to a farnesyl pyrophosphate molecule.
That's a mouthful.
It is.
Okay, so we get this long lipid.
55 carbons long.
Wow, that's a long ferry.
It is.
And this process is super specific, right?
Very specific.
Okay.
The enzyme that makes it creates a specific pattern of double bonds.
So it's not just any old lipid.
Nope. It's got to be just right.
Okay, so we've got our bactoprenol diphosphate.
Then it loses one phosphate.
Why?
Because the next enzyme,
which is?
It needs a monophosphate.
Okay, so we're prepping the ferry.
Exactly.
Okay, how does the loading process work then?
It's a membrane-bound enzyme.
Okay.
It's like the loading dock.
For a bactoprenol ferry.
It brings together bactoprenol phosphate and the UDP Murnac pentapeptide.
Okay.
And it catalyzes the transfer of the Murnac pentapeptide to bactoprenol phosphate.
Releasing UMP.
You got it, UMP.
Other familiar friend.
From metabolism. So now our Murnac pentapeptide is on the bactoprenol phosphate.
Forming lipid A.
Yes, lipid A.
Okay, but we're not done yet, right?
There's more. There's always more.
This is microbiology.
Right.
Lipid A still needs another sugar.
To complete the pair?
The disaccharide unit.
To build that PG backbone.
Exactly.
We need GLCNX.
We do, and that's where MERG comes in.
Another enzyme.
Another membrane-bound enzyme.
Okay. What does it do?
It attaches that final GLCNX
to the Murnac of lipid A.
Yes.
Creating lipid 2.
Lipid caught, the fully assembled building block.
Ready to be flipped.
To the outer face of the membrane.
So our bactoprenol submarine is loaded up.
Yeah.
Ready to cross the membrane sea.
But how does it get across?
Yeah. How does it actually flip?
That's the mystery.
Really?
We know it has to flip.
Right.
But the exact way it happens is still being researched.
Wow.
There's a protein called MERG.
MERG.
It seems to play a key role.
So MERG is like the engine of the submarine.
I like that analogy.
Powering it across. And once lipid 2s is on the other side,
then what?
It can finally be incorporated into that PG layer.
Okay, I think I'm ready for the final construction phase.
Hold on. Before we move on, let's appreciate the bactoprenol cycle.
Okay. What's so special about it?
Well, it's not just for Peptidoglycan.
Really?
It transports other molecules too.
Like?
Components of lipopolysaccharides.
Oh, wow.
In gram negatives.
It's a busy molecule.
It is.
So efficient. I wish I was that efficient.
And here's the thing.
What?
This cycle happens thousands of times.
During?
Each cell division.
Oh my gosh.
Think about that coordination.
That's amazing.
And that precision.
It's incredible.
Makes you realize how complex this is.
It does.
All these microscopic processes.
It makes you think if we mess with that bactoprenol cycle, that could really impact the bacteria.
It could. And some antibiotics do just that.
Really? Like which ones?
Tunicamycin.
Tunicamycin. Okay.
It targets specific steps in that cycle.
So by understanding these details, we can develop drugs to disrupt this.
Exactly.
That's why this deep dive is so valuable.
For you guys.
So, for medical microbiologists like us.
Now, are you ready for the final stage?
Let's do it.
It's time to see how those glycan strands get linked together.
Yes.
And those peptide crosslinks are formed.
I can't wait to see how the fence is built. We're at the final stage of peptidoglycan construction.
The grand finale.
I'm excited to see how it all comes together.
So we have our lipidoglycan building blocks delivered to the outer face of the membrane.
Thanks to our bactoprenol ferry.
Exactly. Now we weave them into that protective meshwork.
So how do they actually get integrated into that existing PG layer?
We have transglycosylases.
Transglycosylases, okay.
T gases.
T gas, okay.
They're like the master weavers.
I like that.
Of the peptidoglycan world.
The tiny construction workers.
Yeah, carefully stitching those strands together.
So they form those beta-1,4 glycosidic bonds.
Yes, connecting the GLCNX and Murnac.
Of adjacent lipid molecules.
Yes, exactly.
So they're extending those glycan chains.
One building block at a time.
Wow, and they do this quickly, right?
Oh, yeah, incredibly fast.
Okay, but we're still missing those peptide crosslinks.
We are.
The ones that give it the strength.
Right, the rivets of our fence.
We need those rivets.
Time to bring in the transpeptidases.
The T-passes.
The T-passes.
They're famous.
They forge those strong connections.
Between?
The peptide chains.
Okay.
So they catalyze the formation of peptide bonds.
Between the chains of adjacent strands?
Yes.
This cross-linking is essential, right?
It's absolutely essential.
For the structural integrity of the whole layer.
Without it, the PG layer would be weak.
Okay.
And easily disrupted.
And the bacteria would be
vulnerable
to all those threats.
Yeah.
This is why TPASs are the targets of beta-lactam antibiotics.
Like penicillin.
Penicillin, right.
Beta-lactams, they bind to those TPASs and they stop them from forming those crosslinks.
So the PG layer is weakened.
And the bacteria can't take the pressure.
Right, the osmotic pressure.
They lies.
Another example of how understanding these molecular details can lead to amazing drugs. But wait, there's more, right?
Always more.
The extent of cross-linking can vary.
It can.
Depending on the bacteria.
Yeah, and the condition.
So it's not just linking every single peptide chain.
Not necessarily.
Okay.
Because the degree of cross-linking, it affects the rigidity and the flexibility of the PG layer.
Oh, so like weaving a fabric.
Exactly.
You can have a tight weave or a loose weave.
And bacteria can adjust that
based on
the environment.
Wow.
Whether they need to be stiff or flexible.
That's incredible.
It affects their survival.
Okay.
Their susceptibility to antibiotics.
Really?
Even their ability to cause disease.
It's amazing how much this seemingly simple structure can do.
It's a marvel of bacterial engineering.
It really is. I feel like we've just been on an incredible journey.
We have from the cytoplasm
to the membrane.
To the assembly of this amazing PG layer.
And now it's time to test your knowledge.
Remember those exam questions.
Get ready to think.
Don't worry, we'll go through the answers.
All right, question 1. Describe the basic structure of the peptidoglycan layer.
Including the composition of those glycan strands.
And the types of peptide crosslinks.
In both gram-negative and gram-positive bacteria.
So the peptidoglycan layer is that mesh-like structure.
Right.
It surrounds bacterial cells, giving them support and protection.
It's like a chain-link fence.
And the glycan strands are those repeating units.
Of GLCNAC and MRNAC.
Linked by those beta-1,4 bonds.
Yes.
And then those strands are connected by the peptide crosslinks.
Which are different in gram-negatives and gram-positives?
Right, in gram-negative.
Yes.
The crosslink is usually a direct bond between DAP and D-alanine.
Exactly.
But in gram-positives, it's a lysine linked to another peptide chain through a bridge of glycine molecules.
Often a pentaglycine bridge.
Making it longer and more flexible.
You got it.
Okay. Question 2. Explain the role of bactoprenol.
In PG biosynthesis.
Highlighting the different stages where it's involved.
And its significance as a potential antibiotic target.
Bactoprenol are molecular fairy.
Yes, it's essential.
It shuttles those building blocks across the membrane.
From the cytoplasm to the outer face.
And it's involved in several stages, right? Yeah. First, it picks up the UDP-MRNAC pentapeptide.
In the cytoplasm.
Then transports it across. And then it's incorporated into the PG layer.
Exactly.
And because this cycle is essential, disrupting it, it can kill the bacteria.
Makes it a good target for drugs.
Like tunicamycin.
Tunicamycin, yes, which targets specific enzymes in that cycle.
Exactly.
Question 3, discuss the 3 phases of peptidoglycan biosynthesis.
Okay. The cytoplasmic.
The membrane.
And the extracellular.
Oh, okay. So in the cytoplasm, we assemble that UDP-MRNAC pentapeptide building block.
From simpler precursors?
Like glucose and amino acids. Using those MER enzymes.
URAF, right?
Yes, and alanine racemase.
And D-allyligase.
You got it. What about the membrane phase?
The membrane phase is where bactoprenol comes in.
Our star player.
We transfer that UDP-MRNAC pentapeptide
to bactoprenol phosphate.
Creating lipid E's.
Yes.
And then we add GLCNAC to form lipid 2.
Using MER.
And then that flipase.
The mysterious flipase.
Flips lipid 2 across the membrane.
Okay, and the final phase.
The extracellular phase. That's where the action happens.
The PG layer is built.
Lipid 2 is incorporated.
By T gases.
And cross-linked by T passes.
Perfect. You aced it.
Okay, what is the significance of D-alanine?
And how is it made in bacteria?
Okay, D-alanine, it's essential for that transpeptidation reaction.
With cross-linking.
The D-alanine.
That may end of the peptide chain.
It's recognized by T passes.
And the terminal D-alanine is released.
During the cross-linking.
And it's generated from L-alanine.
By alanine racemase.
Which is the target of cycloserine.
Right, cycloserine inhibits D-alanine formation.
Disrupting PG synthesis.
Okay, last question. How do cytoskeletal elements contribute to PG synthesis and cell shape?
Okay, this is about those internal scaffolds.
Yeah, like those actin-like proteins.
And they guide and regulate PG synthesis.
They direct where those syntheses go.
Ensuring new material is added in the right spot.
Which determines cell shape.
Like in rod-shaped bacteria.
They're made
which helps direct PG synthesis along the sidewalls.
To make it elongate.
Exactly. They're like architects shaping the cell.
Wow. I feel like I've learned so much.
Me too.
This deep dive has been amazing.
It's been a journey.
And this knowledge is so important for medical microbiologists.
Absolutely.
It's not just for exams.
It helps us understand how bacteria work.
How they interact with us.
And how to fight infections.
So before we go,
yes,
one final thought to ponder.
Okay, I like this.
We've talked about targeting the bactoprenol cycle and inhibiting T-passes.
Right.
But what other steps in PG biosynthesis could we target?
Ooh, that's a good question.
What about those enzymes in the cytoplasm?
Or the flipases.
The flipases, yeah.
Or even the cytoskeletal elements.
It's exciting to think about the possibilities
yes.
for new therapies.
New antibiotics.
Maybe one of you listening will discover the next big target.
Maybe.
Thanks for joining us on this deep dive
into Peptidoglycan.
Be sure to check out the show notes for more resources.
And we'll see you next time for another microbiology adventure.